Good afternoon, everyone. This is Suzanne Vernon, the Scientific Director of the SolveCFS Initiative. Thank you very much for joining us today for the second of our webinar series. We are very excited to have Patrick McGowan presenting today. Patrick is one of our 2011-12 funded investigators. He is an assistant professor at the University of Toronto, and he's going to be talking to us today about his research that he has been conducting for the past 2 years on the epigenetics of ME/CFS. Before I turn the controls over to Patrick, I just want to give you a little bit of information about how Patrick kind of came into the field of ME/CFS research. So a couple of years ago, as I was trolling the literature and doing what I like to do, trying to connect the dots of important research for ME/CFS, I was reading a paper that was extremely interesting. It's about the HPA axis and cortisol and how the environment can impact the actual control of the HPA axis. This turned out to be a very important seminal paper, if you will, that Dr. McGowan actually published. And so I sent Dr. McGowan an email. And I said, hey, how would you be interested in maybe thinking about applying your incredible research knowledge and expertise to the study of ME/CFS? And he responded back and he said, that sounds fascinating. ME/CFS sounds like an incredibly important, complex, intriguing problem, just ripe for scientists like Dr. McGowan to tackle. And then he applied to our funding opportunity that we issued in 2012. And obviously his application was very, very well received, and has resulted in the research that he's going to tell you about today. So I'm going to right now switch controls to one of the 300 people that are listed. Let me see if I can find Patrick on here. Oh my. Bear with me here, folks. I have a list of all of the presenters. There you are, Patrick. So hopefully the controls will now be passed over to you. Is that correct, Patrick?
That's right. Yeah, I have the controls, and you should be seeing my screen at this point. So, all right. Well, thanks very much, Suzanne. That was a great introduction. I really want to start out by thanking the SolveCFS ME Initiative and for all of their support and taking the chance to fund me and to bring me into the world of ME/CFS research. So I want to say, I'm a basic research scientist, meaning that I don't work directly with patients. I don't make diagnoses. I am primarily involved in collaborations with people who do that kind of work, and our interest is in understanding the biological bases of a variety of complex diseases. And CFS is among them now, and we're very glad for that. I'm also very thankful to the people who are attending this seminar for their interest in our research and your support. From the perspective of people donating their time, their money to helping us to understand the biology of ME/CFS. We're very grateful for that. The research that I'm going to discuss today, I'm going to try to provide you with a context so that you can understand the recent work that we've been doing, in particular some of the work that we've just published on the epigenetics of ME/CFS. A lot of this work was done by Will de Vega, who's a graduate student doing his Ph.D. in cell and systems biology at the University of Toronto. And we've teamed up with Dr. Vernon to explore the issue of the epigenetics of CFS in a way that allows us to ask about how the environment and genetics are, the interaction between the two are embedded in our biology. So let me give you a brief overview of things that most of you are very familiar with. So obviously ME/CFS is under the category of complex disease. So in other words, this is a disease that affects a variety of different physiological systems. This has been part of the issue for researchers trying to understand what it is that what are the biological consequences of ME/CFS. Because there is a heterogeneity in the population of sufferers. So some sufferers will present with more of some of the so-called phenotypes associated with CFS, and some people will present with others predominantly. So what we did in our research is that we developed a study that would allow us to look in an unbiased way at biological markers in a population that was quite well controlled. So we started with a relatively small group of individuals who were CFS sufferers and had donated their blood cells to a variety of different biobanks, and as part of the SolveCFS BioBank. So our samples come from that pool. We also have healthy control individuals to compare the biology that we see that's different in the ME/CFS sufferers. So what I study primarily is epigenetics. So epigenetics is a relatively new field of research. There has been a huge effort in biology to understand how genes impact chronic disease. And one of the things we understand, of course, is that although every cell in our body has virtually the same genetics that we inherit from our ancestors, not all cells behave the same way, right? Some cells are red blood cells, some cells are muscle cells, some cells are brain cells. So how do they achieve that difference? Well, they do so by epigenetic processes. So these are processes, the word epigenetics actually means above the genome. These are biological processes that interact with the genes, that change the way the genes function, but don't change the genes themselves. So they don't change what's passed on to your children and your children's children, but they change the way these genes work. And so each of the different types of cells in our body has a very different epigenetic profile. So another way to talk about it is that one could think about our genetics or inherited genes as the hardware of our body. So these are the things that, these are the sequences of A, T, C, and G, our alphabet, that programs the basic development of our body and essentially provides the platform that lets our body develop into what it is. The epigenetics is more like the software, so it runs on the genetic program, but it can take a variety of different forms. It can do very different things. So the epigenetics then are really biological processes that control when, where, and how our genes work. So obviously very important for understanding when things go wrong. What are these how are these processes affected? I'll give you an idea of how this has been influential. So there have been a number of studies throughout the decades looking at identical twins, because identical twins have the same genome essentially. But often, in fact very often, when it comes to complex diseases, they show very different phenotypes. So if you just look at the chart on the right, it's just showing you a variety of different complex diseases, from diabetes to rheumatoid arthritis to autism. And you can look at the concordance. So if one twin has the disease, what is the likelihood of the other twin also having the disease? What you can see is that there are some complex diseases where you see a lot more concordance between identical twins than between fraternal twins. So fraternal twins, they share the same womb, but they don't have the same genetics. They share about the same genetics as brothers and sisters do. So often what you see is that the identical twins will share more of the same complex diseases, but it's never 100%. If genetics was the cause of these complex diseases, one would expect that, in fact, 100% of the time you would see if one twin had the disease, the other twin would have it as well, but you don't see that. So people have started to look at epigenetic tags. So these are additional processes I was telling you about. And if you look at twins, on the left, what it's just showing you an example of a chromosome, where you can see that it's been stained to show you where the epigenetic tags are similar and where they're different. You see that there are a lot of similarities in very young twins at 3 years old. So these are the yellow marks where the twins have the epigenetic tags in the same place. As twins get older, they show more and more differences in their epigenetic tags. So epigenetic tags are susceptible in some senses to environmental influences. This is our hypothesis.
Hey, Patrick.
Yeah, sure.
Can you use your cursor? Yeah, that would be great. That would be great.
Absolutely. So a lot of the work that, a lot of the ways that we know that how the environment interacts with our genetics come from studies in animals. And this is just one example. So these two animals, for example, these are two mice, lab mice, are genetically identical. So they're like clones of each other. But obviously the phenotype is very different. So one mouse gets really fat, susceptible to cancers. The coat color is different. That's a dead giveaway. The difference between these two mice has to do with the amount of nutrition that their mother got that contains some of the bases for modifying the epigenome. So this kind of study launched a lot of interest and investigation into trying to understand the epigenetic differences that are associated with complex diseases in humans. And without having a degree in biochemistry, to put it simply, what we're looking at is we're looking at a modification of the DNA itself by adding a methyl group to the DNA. So DNA methylation. That's the epigenetic tag that we're looking for when we look for these epigenetic differences.
Patrick, can you give a brief explanation of what you mean by phenotype?
So the genotype is your inherited genetics. And so this is the sequence of letters of the alphabet that make up your all the DNA that you inherit from your parents. That's your genotype.
And phenotype?
Phenotype is the function of your genes, so how they work. And phenotype can also be what you look like. So how you behave, how your brain works, how your body works. That's your phenotype.
Thank you.
Sure. So when we talk about how an understanding of how our genetic variations, so our inherited DNA, and the exposures that we get from the environment interact, epigenetic variation really sits at the interface between the two of them. So the environmental exposures and the genetic variation that you have are reflected in your epigenetic variation, these marks on the DNA. And it's those marks that we're looking at when we're in this study that we did of CFS. One of the interesting things also clinically about your epigenome as opposed to your DNA sequence, your genetics, is that as I said, they alter the way your genes work, but they don't alter the way your genes that are passed on, your inherited genetics. The epigenetic marks are potentially reversible, unlike genetic mutations. So if you have exposure to radiation, for example, that could cause mutations in your genome, and your genetics would be altered. Your epigenome, but in almost every other case, your genetics are what you're born with and what you will die with, so they do not change. The epigenetics that you have, however, are potentially reversible. So a number of different kinds of interventions have been shown, and particularly in animal models, to reverse some of these marks. So that opens the potential for alternative or novel therapies that can target epigenetics as a way to modify pathology associated with chronic fatigue syndrome. So what can we do about our epigenome? So as I said, there are a number of different ways that the epigenome can be modified. Some of the most advanced research in this area is being done in cancer biology. And in cancer biology, there's scores of pharmacological therapies that have been approved by the FDA that target epigenetics. Just one example is 5-azacitidine, and that is a drug that alters DNA methylation. And that drug has been approved for treatment of some cases of some types of leukemia, so blood cancer. Other potential therapies are less well understood. However, there's a lot of interest in this. So there's a lot of talk about how modifications to diet, other kinds of interventions like meditation, for example, might modify some of the genes that are involved in laying down these epigenetic tags. The evidence on this is right now lacking. So there are some studies, but there needs to be a lot more studies to understand what are the potential therapies that can be effective to alter epigenetics and when they can be effective, because it's likely that they're not going to be effective at all stages of life. So I'll go into our study goal now. So to give you a broad overview of what we've done in our study, as I said, we've taken advantage of the fact that the SolveCFS BioBank has a resource of samples from CFS sufferers and from healthy controls who've donated their blood cells. We've used the latest technologies to look at the epigenome, which is called a microarray. It's a DNA microarray, so that we can categorize, we can look at the entire genome and look at all of look at a number of different marks, different DNA methylation marks that are modified in CFS, that are different in CFS compared to healthy control individuals. And then we can group these genes into pathways to understand how they could be related to physiological differences in a number of these different physiological systems that are affected in CFS. So in the study, we had 12 highly selected CFS patients and 12 healthy control subjects that were matched along a number of parameters. In this case, we chose all females. We have a lot more samples from females than males, and we expect that there's likely to be sex differences in the way that people present CFS and the physiological systems that are impacted by CFS. They were matched for age, they were matched for BMIs, so body weight. So in this sample, we didn't take anyone who was obese, so BMI over 30 for this study, because obesity has been shown to influence blood cell inflammation. And since we're looking at blood cells, we didn't want that to vary as well. And what you can see on this chart is the results of the RAND36 information about physical functioning and mental health. And what differed among these subjects is they showed large differences in physical health, statistically significant ones, between the CFS patients and the control subjects. We didn't see a difference in mental health in these samples. So, as I said, we're looking at DNA methylation. So what's the role of DNA methylation, this mark on the genes? How does it affect the way genes work? At a very basic level, one could think about DNA methylation as like a dimmer switch on a gene. So when there's an unmethylated gene, the gene is generally switched on and produces RNA and produces whatever it's going to produce in the cell, whatever product. If the gene is highly methylated, if there's a lot of methyl groups attached to that gene, the gene is generally switched off. And depending on how much methylation you have there, you have different levels of the expression of the gene. So the gene is sort of switched off or switched on depending upon this methylation mark, the presence of it. And we're looking at different regions of the gene here. So without having to know, if this would be too complicated, you can ignore it. But for those of you who do know, genes have different, there are different parts of the gene that determine how it works. And in the front of each gene, there tends to be a regulator of the gene. So we can look at the methylation in these regulator genetic elements, and we can look at the methylation within the genes as well, in the body of the gene. And we think that they do different things, which is why we're interested in looking at where the methylation is. So the paper that we just published in PLOS ONE, we just wanted to categorize how the epigenomes were different in these two populations that we're looking at. So where did we see the most differences in DNA methylation? And it turns out that the regulators of the gene, those parts in the front of the gene that determine how the expression occurs, were some of the most altered in terms of their epigenetic profile. We then looked at grouping genes together with similar functions, and we wanted to say, okay, we found, say, 1,000 different differences across the genome in epigenetic marks. How many of these genes showed epigenetic differences how many of these genes do similar things in the ones that showed these differences in epigenetic marks? So we grouped them together by functional categories, and we found that genes that regulate how the immune system work were among the most affected epigenetically in our study. And there were also other types of functions of genes that were affected predominantly in the CFS patients compared to the healthy controls. Ones that relate to kinase activity, to just the basic components of how the cell works, and cellular metabolism. So this information supports a number of other studies in the literature that have shown that the function of immune genes are altered in CFS. But what it does is what it adds to is that it says that one of the reasons potentially that you see these alterations in the immune system function is because of a change in the epigenetic marks on the genes. And we think that those epigenetic marks are very stable, so they don't tend to change very much in the face of daily life exposures, daily life events. So we think we may be looking at least in part, some stable biological processes that could alter the way genes work in the immune system in CFS.
Hey, Patrick.
Uh-huh.
Oh, actually, let's finish this last slide. I'm getting a lot of questions about the basics of methylation and
Sure.
and what that is, what that means. So maybe once you finish this slide, we could go back to your slides that again explain what methylation is and how that could affect the activity of the gene and what it did to those genetically identical mice.
Sure, absolutely. Yeah.
Okay.
I think this is my last data slide. So
Okay, great.
it's a good point to do that. So to wrap up, in our study, what we did then is that we took all of these genes where we saw differences, and we said, okay, where are these differences in epigenetic marks with respect to the structure of the gene? So were they in those regulator areas, or were they in the bodies of the genes themselves? And what we found is that the differences were not random. The take-home message here is that the differences that we saw were not randomly displaced throughout the gene, that they happened to cluster in particular regions of the gene. So we found the greatest effects in the immune genes that we found were in the regulator elements rather than in the bodies of the genes. So we think that that will help us in the future to identify why it is that those genes, the function of those immune genes, are altered. So do you want me to go back now to the I think I had one conclusion slide, but I can go back.
Yeah, I think go back. Am I still on?
I can hear you.
Okay, good. Yeah, I think go back and just kind of review again. So now that we know all of these changes about that are happening in ME/CFS samples, let's just review again what this methylation is and what it does.
All right, let's go back to. This is great. It's very interactive. I like that. Um, so what is methylation and what does it do? So as I said at the beginning, what you're born with is you're born with your genetic hardware. So the genes you inherit from your parents that determine, it's like a basic blueprint for building your body and building your brain. The epigenetic marks on your genes are critical and necessary for this as the software in your body. So it tells the genes how to work and when to work and how much genes, how much expression to produce in which kinds of cells, in which organs of the body.
So walk through those really cool illustrations that you have there. So you have a computer, that's the hardware, and then.
And then, so for example, yeah, your computer is your hardware, but it doesn't really do anything unless you have the software, your system to run it, right?
Right.
So that system is the epigenetics. That's the software, the programs that you're running on your computer, your hardware, right? So on your iPod, you got an iPod. The music that you download is the software. That's the epigenetics. So that actually is what you're listening to. But the basic thing that allows you to listen to the music is the hardware. So that's your iPod, right?
Perfect.
Okay. So what we know about how epigenetic marks differ in humans compared to genetic differences comes from some studies of identical twins that have been really illustrative of this, because identical twins have the same basic blueprint. They have the same genetics, same inherited genes. But it's well known now, it's well understood that even from birth, identical twins can have different epigenomes. So depending on how different the identical twins are at birth, so some twin pairs are born with very different weights, for example. That's very common, even though they have the same genetic blueprint. The greater the weight difference, the greater the epigenetic difference. So what this tells us is that the epigenetic differences are associated with a difference in how the genes are working. So the phenotype, and that creates the phenotype of the individual, right? And some of the coolest research that has helped us to understand how the environment can influence how genes work through epigenetics comes from animal work. We don't have a clear understanding very often about how environment can interact with our epigenome in humans, partly because we can't access a lot of the time the right tissues. It's very hard to control those studies. There are ways to do it that we've been thinking about for ME/CFS, but often the basic science is done in animals, and some of those studies have indicated that at some points in life, at some stages in life, nutritional interventions or pharmacological interventions can alter the epigenome and also alter the phenotype of the animal.
It's very, very helpful, I think, for helping us understand this very interesting, but yet also complex topic. Do you think epigenetics is really what adds to the complexity of ME/CFS?
I missed something you said there.
So epigenetics is probably one of the factors that really contributes to the complexity of ME/CFS.
It's likely that it does. I mean, we think about epigenetics as this interface between the environment and our genes. And it's, until quite recently, epigenetic processes have been largely ignored in research because it's been assumed that you basically are born with your epigenetic profile and it's similar throughout your life. But there's been a lot of research in the last 5 years. In fact, it's been very recent, but there's been a lot of it showing that, in fact, your epigenome is susceptible to change throughout your life. And so now what we have to understand is what's causing the change. In our study, we were starting with the idea of just looking at what's different, and without making an assumption about what's causing the difference in CFS-ME, right? So if we can understand the epigenetic differences, then I think we have a greater understanding of that genetic environment interaction that leads to this complex disease.
That's great. Thank you so much, Patrick, for going back and reviewing that with us. Now,
sure,
maybe
absolutely.
if you could jump to the conclusions. The questions are piling up here, so okay, I'm going to leave time for some of the questions.
Well, let me just wrap up quickly then to talk about the conclusions here. So, in our study, what we've done essentially is an exploratory analysis, and the goal of which is to identify biological markers for CFS in terms of epigenetic tags. And what we found was that a number of genes have different epigenetic tags that are related to the immune response and to metabolism and to kinase activity. So there are a number of processes that seem to be affected epigenetically. The data are really consistent with other evidence that ME/CFS really involves a multi-system dysregulation, and it implicates epigenetic processes, specifically DNA modifications in the pathology associated with ME/CFS. So our next step, we're very interested in twins as well as a way of understanding the epigenetics in the absence of any genetic differences, and we've started some studies on a few sets of twins. We'd love to look at more. Right now we only have four twins to look at that are monozygotic. We're interested in some of the physiological differences associated with ME/CFS, such as HPA axis dysfunction, which is a regulator of all kinds of basic physiological processes, from circadian rhythms to stress. We're interested in the association of those kinds of changes with epigenetic differences. And we're interested in interaction of CFS with other conditions that are associated with CFS, such as obesity. Now we can go and we can grow our subject population and look at a larger number of samples with other kinds of physiological presentations associated with CFS, and we can start to try to understand, can we subtype groups of CFS sufferers based on their epigenetic profiles. So thanks a lot.
That's great. All right, I have many questions, so I'm going to start from the top, Patrick. Mm-hmm. You addressed this one very briefly, but I'll just read it. Is there any research about the effects of meditation, either daily or long retreats, on epigenetics?
There are very, very few studies on this. Some of the best research is done out of University of Wisconsin-Madison by Richie Davidson, and he studies long-term meditators, like expert meditators. And there is one study that he did last year that showed changes in the function of some genes that are change epigenetic profiles in long-term meditators, as opposed to with an intense meditation. So it's one study, and it's just a very small piece of information. But clearly, it's something that we need to follow up on. I don't think we have enough data yet to really say definitively that these kinds of interventions can have a major or a long-term impact on epigenetics. And we really don't know enough about how it can, what are the conditions that you need to actually impact the epigenome. It's not, so I want to say that what's important to understand about epigenetics is that what we're not saying is that one can sort of think their way out of a complex disease in many cases. In other words, you know, and this is something that you see on YouTube sometimes, is that people talk about epigenetics as though one can cure cancer by thinking good thoughts and positive feelings and that kind of thing. There's absolutely no evidence for that. But there is evidence that there's some environmental condition interventions, pharmacological interventions can alter the epigenome, and that can have an effect on some complex disease.
Very interesting. If the gene is hypermethylated, do you want it to be unmethylated to correct expression?
Right. So that's what you would want to do, is you'd want to be able to selectively target your gene, that's your problematic gene that's hypermethylated, and you want to unmethylate that gene. The difficulty is that even the best kind of interventions that we have, which are pharmacological, that have been used in cancer therapies, are very dirty drugs. So they affect a lot of different things. And we have yet to find a way to specifically target one gene of interest. But there's a lot of research into that. That's basic research that's asking that question. How do we target specific epigenetic changes? I think one way to think about how you might get at that question is that there are a number of interventions that we already know that have a positive influence and impact on CFS, for example. And you know, it's likely that part of the way they're doing that is that they, you know, for whatever reason, they're affecting the right kinds of genes, the function of the right kinds of genes. And so we have to understand the epigenetic signature of some of those interventions. That would be a really cool step to take in this research.
There's a question about, is there a way to get a copy of this study? And we have a link to the paper describing the results that Dr. McGowan has just presented on our website, SolveCFS.org. Patrick, where do you foresee your research going in the future? And how could it potentially, where do you foresee your research going in the future? And how could it potentially shed light on the processes of CFS, the mechanisms of CFS, and possible treatments?
Well, you know, as many know, there are no biological markers of CFS. So what I hope with this research is that what we will be able to do is identify biological markers that can be used to gauge the effectiveness of interventions. I'd like to see how stable these marks are over time, and in the face of interventions as well. And so I'd like to look at this in larger numbers of subjects. I'd like to look at individuals who have a variety of different kinds of presentations of CFS in different, for example, people who show obesity and also CFS. You know, these data can be used potentially to subtype people with different presentations or different physiological manifestations of CFS. And so ultimately, I think, you know, from that perspective, I think that could be useful, because it would provide a way of diagnosis that we don't have. And it could also, as you know, as I said, pharmaceutical companies are racing to understand the impacts of epigenetic drugs. So it could potentially open new avenues for pharmacological treatments that don't exist right now for the treatment of some of the pathology associated with CFS.
Great. So there's a question about, does methylation happen through environmental exposures such as different types of diets?
So methylation, so much of our methylation that we have in our genome is there from birth, because as I said at the outset, you know, you have different types of cells, and those cells have different epigenetic profiles. So if anything could alter your DNA methylation, if any kind of environmental event could alter it, you'd have major problems, right? So your skin cells would become blood cells, would become muscle cells, and they don't do that. So there's a lot of protection of the genome, of the epigenetic marks in the genome. But at the same time, for some genes, in some cases, in some types of cells, the DNA methylation can be altered by the environment. And so this area of research is relatively new, but there are a number of studies trying to identify what are those conditions to alter it. And I think we still don't have a great understanding of what those conditions are. We know them in some cases, but not. We don't know enough about the variance in epigenetic marks and how they can vary throughout the lifespan.
I didn't realize that. I actually thought that epigenetics, methylation in particular, was a lot more dynamic than that. But so yeah, that was very, very interesting. How did your study, or did your study, reveal the level of gene expression of interferon genes in ME/CFS?
Well, we didn't look at gene expression in this study. And other studies have done that. We'd like to do that. We'd like to combine looking at epigenetic marks with looking at the expression of the genes. But I'd have to go look at our list. But I did want to say, related to the publication of the paper, is that this paper is open access, so anybody in the world can go and look at this paper. We'll have to provide a link to it, but it's published at PLOS One, and you can find it. If we'll make it hopefully we can make a link available to people, but we've basically deposited all of our information so it's freely available, including all of the raw data, and you can find a list of all of the genes that we found that were differently methylated. I'd have to look to see specifically interferon. I can't recall if that was one of the ones on our list.
And we do have a link to the paper on the website, and as Patrick said, it is an open access paper, so you can go to it, you can download the PDF, you can download all the supplemental material, openly and freely available. This is a very interesting question. How would a disease such as CFS cause one's epigenetic marks?
Well, at this point,
Speculate,
we know a little
maybe.
something. Yeah, to speculate about how it would alter epigenetic marks. Well, we were agnostic about the causal element in the CFS equation in this study. We wanted to not bias our results. We wanted to sort of just look across the entire genome and all of these changes. But at the same time, we know that some environmental impacts can alter some of the physiological systems that we see that are altered in CFS. So by extension, like if we look at, for example, infections have been shown to alter the epigenome. So, and they don't just alter random genes, but they alter genes within the immune system. And we did see differences in epigenetics marks in immune system genes, important immune system genes. Now, one of the future goals that we can of our work would be, since we've deposited all this information and all the raw data online, a number of other studies are doing the same thing. So what we can do is we can go and look at those studies and say, okay, can we now compare the epigenetic profile in conditions where you have infection with the ones that we saw in our case, and do we see a signature of that in the epigenetic modifications that we have? And as we get a larger sample size, we can start to do that in a more comprehensive way. But that would be one way to approach that, to try to compare across different studies by looking at all the raw data. This is one of the nice things about this kind of genomic work now, is that a lot of the time we have the information available online now, and there's been a lot more sharing among researchers of that information. That will be very helpful to us to answer those questions.
There's a lot of questions about the impact of infection on epigenetics. What do we know about that, if anything? Or is that also a pretty new area of investigation?
It's relatively new, but there are a lot of studies. So one of the things that we didn't do in this study is we didn't look at specific cell types within the blood. We looked at all blood cells that had a single, they were called mononuclear cells. They have a single nucleus in the cell. And that constitutes a variety of different kinds of immune cells. But there's a lot of basic research that has looked at the epigenetic profiles of those subtypes of cells. So again, what we can do is we can go and look at that research, and we can compare the profile of those cells that are altered with infection at a specific cell level, and we can compare that to the data that we've generated to see, do we have evidence that some of those cells in our sample are also epigenetically altered. That will give us more of an indication of what kinds of cells to look at. But the reality is that in ME/CFS, in the literature, there are a number of different cell types that are affected in the immune system, not just one. So I think that's hopefully one of the things that this research can do is it can help to be a useful tool to start to ask questions about the impact of infections on epigenetic profiles and cell function. But I think we're really at, kind of at the beginning of that. We have to integrate our data more with other basic research.
Sounds good. I think this will be the last question, but it's kind of connected to what you just said, Patrick.
Mm-hmm.
What other groups of affected genes could have been detected than those that affect the immune system? I mean, how did you group genes? How many groups were there roughly?
Right. So what we did is we used, we looked at all of the data out there in the literature on what each gene is doing. So a gene can have multiple functions, right? A single gene. But if you combine a number of genes together, you can see that most of them are involved in one function or another, right? So, for example, we found, we're looking at blood cells, and we found that the immune response genes are particularly affected. But we also found that there are a number of other classes of genes that showed predominantly more differences than others. And these are genes involved in cellular metabolism, so how the use of energy by the cell, and kinase activity, which is how proteins interact with each other in the cell, and basic components of the cell, so the structure of the cell, its cellular membrane, and that kind of thing. So we're using all of the data on what each gene does is deposited in a database, and we can take that, and it gives us a relatively unbiased view of what each gene's function is. So we can look at our data, and we can say, okay, we know from many other studies the association of this particular gene that's modified in CFS with the function of the immune system, or with cellular metabolism, for example, and we just group them together by the dozen to see which are the most prominently affected. So what we showed in the paper was that there are these 4 clusters or 5 clusters of genes that are predominantly affected. There are many others as well, but it gives us sort of a snapshot of, it helps us to identify particular pathways by grouping genes together, because any one gene will have a number of different functions.
And I think that maybe if you could say just a little bit about the fact that many of the genes in the immune pathways had less methylation,
Mm-hmm.
and therefore, what does that lesser amount of methylation possibly mean for the regulation of those genes?
Right. So, so by and large, less methylation is associated with increased expression of the gene. So the gene will pump out more RNA and make more proteins. So by and large, that's what we found in the immune system. And I didn't really talk too much about the direction of the difference, but that's really important. And it's going to tell us something about how the function is, how we expect the function of the gene to be altered.
Yeah, well, this is the first in a number of exciting results that Dr. McGowan has presented for us and you will be seeing in the future. So he is actively digging deeper into many of the issues that you have raised. And again, his research. Patrick, if you could hand over the controls back to me.
Absolutely.
His research was made possible because of you and your generous support. And obviously, because of your participation in the biobank, there are a lot of questions from people about, I live in Canada or I'm in the UK. How could I possibly be part of this study? And our biobank helps you do exactly that. Can you see my screen, Patrick?
Presentation. There you go.
Thank you. So there are a number of ways that you can participate in research, that you can follow what's going on in the field. If you don't, want to give your blood or stuff like that, but still, keep up and be part of the solution regardless. So sign up for our Research First e-newsletter. And as I indicated, sign up for the SolveCFS BioBank. It was because of the biobank and the contributions in samples and information that many of you have made that made this research and what Patrick is continuing to do possible. I mean, this is cutting edge, very, very exciting stuff. This webinar will be posted on our YouTube channel, and you should be seeing that recording posted in the next day or so. Also, we have a number of other webinars coming up. I think we have one every month until the end of the year. So go to our website, solvecfs.org, and register for those webinars, and you'll get reminders and everything else you need to be able to log on in time. Of course, like us on Facebook. The more likes we get, the more the word about ME/CFS gets out there, the more awareness there is about it, and all that helps increase interest from all sectors of research and medicine into ME/CFS, and that will help drive research even further, accelerate it. And of course, research is expensive, so please donate to the organization so that we can continue to support this amazing type of innovative and cutting-edge research like that Patrick has just presented. So we are finished with our second webinar. Thank you very much, Patrick, for a very interesting and exciting presentation. And we look forward to seeing everyone and more on our next webinar. Have a good afternoon.
Thanks very much, Suzanne, and thanks everyone for your interest. Thanks for your support.